All right. Good afternoon, everyone. I need to also apologize for not making it this morning, and unfortunately, I promise immediately after the commitment, I will let you know, and eventually, when I'm about to invite you, another thing comes off, so I have to also go to that. So I decided at least also not to miss the day. So I will do the recording, which immediately after this recording, I will try and communicate with you. So that we can have a little discussion also with you. So I will record this and then I will share it as you go. And I'll also try and update the platform if possible today, so that we can have access to the link. And I will also post some tutorial questions in the platform so we can start our discussion. So please try participate in the discussion. One, number two, I observed some of you also doesn't follow the classes, the recorded classes. Please try and follow the recorded classes so that compare what was given in your lecture material. How do you even explain what was given in your lecture material? Get the explanation. And if you have any question, please do not hesitate to drop it in the chat box or in the discussion. It's unexpected. So today we are going to look, we are still in module 2, chemistry of nutrients, study section 2, chemistry of sugars 2. In our last class, we introduced the concept of carbohydrate, the biomolecules generally, where we talk about the classification, the chemistry, the glucose, the structure, isomerization, formation of cyclic form of glucose. Today, we are going to see, we're going to continue where we'll be discussing basically the the saccharides and polysaccharides. Basically, they are on chemistry rather than chemistry. So before we start that discussion, I felt it is necessary to have an idea, because we will be talking about some monosaccharides that you are not too familiar with, at least at some point. So I need to tell you the concept of how this monosaccharide comes from, and why do we have them instead of the normal monosaccharide we mentioned while we're discussing monosaccharide, that the ketone or ketosis or the aldosis. There are some monosaccharides, or the same monosaccharide can undergo some modifications to produce what we call, or to form what we call derivatives. They are derived from those monosaccharides we mentioned. So there are many. So just to mention a few, and in our process, in the process of the discussion, we will be talking about some of them so that it will not appear strange for you, so that you have an idea where they come from, where does that come from. Okay, starting with sugar acid. We have what we call the sugar acid. So It is monosaccharide that undergoes some reaction to produce a modified form of monosaccharide. So it can produce a sugar acid. What is this sugar acid? When oxidation of aldehyde or primary alcohol, for example, in aldosis, when you have oxidation of aldehyde or primary alcohol, for example, in glucose, we have glucuronic acid or glucuronic acid or gluconic acid, depending on where the oxidation takes place. If the oxidation takes place in the aldehyde, if the oxidation takes place on the aldehyde carbon, that is carbon 1 of glucose, which form what we call gluconic acid. But if the oxidation takes place at carbon 6 of glucose, we form what we call glucuronic acid. That's the point where we discuss we mention some of this as form of carbohydrate. Polymer, where we mention uronic acid instead of glucuronic. So basically, in a uronic acid, and this is just an example of the case of glucose. There is galactose, and that can be galactonic or galacturonic. Same thing with mannose and the rest. So when we mention uronic, we mean the one where oxidation takes place at carbon 6. And it could also be, when we say uronic is general, it tells that the oxidation takes place at carbon 6. It could be galacturonic, mannose, glucuronic, maybe other one. Yes. And the next one, we have sugar alcohol. And in sugar alcohol, it is the reduction of either the aldehyde in the case of aldosis or ketone group in the case of ketosis to produce a sugar alcohol like sorbitol, mannitol, ribitol. So when glucose is reduced, it forms sorbitol. When mannose is reduced, it forms mannitol. When ribose is reduced, it forms ribitol, etc. And the next one, we have amino sugars. In amino sugars, when we have one or more of the OH group, you know that the glucose, monosaccharide contain a lot of OH group. So in a situation where by one of the OH group is replaced with an amino group, for example, in glucose, it can be glucosamine. If it is beta D glucose, it will be beta D glucosamine. If it is galactose, it will be beta D galactosamine. If it is mannose, it will be beta D mannose-amine. Indicate that an amino group is attached to it, but it is amino sugar. There are some cases where an amino sugar can be acetylated, an acetyl group can be added. In that situation, we'll be having N-acetyl. So if it is, for example, beta D glucosamine, and it is acetylated on the amino group, that meaning an acetyl group is attached to the amino group. It is called N-acetyl beta D and beta D glucosamine. There are some other cases also like muramic acid. In muramic acid, what happens is it is glucosamine then added or reacted with a lactic acid. There is also rhamnose. There is, which are the oxy sugars, which you've already explained what happened in the case of the oxy sugar. When sugar is reduced at one of the OH group, like we give an example of the oxyribose, and we have beta L-fructose, which is 6-deoxygalactose, and we have alpha L-rhamnose, which is 6-deoxymannose. So in case, please, we mentioned this, you should be familiar with these derivatives of sugar. Okay, now discussing disaccharides. We've already introduced what disaccharides are. And the simple explanation we give is that in disaccharide, we have two monosaccharides linked together by the type of bond we call glycosidic bond, or glycosidic bond. And I think we've explained why the O. So usually they form when the hydroxyl group of one sugar reacts with anomeric carbon of the other sugar, even though we can see some variation along the but what is general is that these disaccharides, by physical properties, they are usually crystalline in nature, sweet, and they are water soluble, just like the table sugar. See what constituent of the table sugar shortly. So generally, disaccharides are divided into reducing and non-reducing. So there can be reducing disaccharide or non-reducing disaccharide. What are reducing disaccharide? Why are they called reducing disaccharide? Reducing disaccharide when you have free anomeric carbon. Remember, we mentioned what is anomeric carbon, the carbon that determines the alpha beta configuration. Yes, that was formed after the cyclization, which is the anomeric carbon. So anomeric carbon is usually carbon 1 in the case of aldosis and carbon 2 in the case of ketosis. So in disaccharide, you have two sugar forming a bond. So the two sugar have anomeric, each one have its own anomeric carbon. If the bond formed between the two sugar involves both the two anomeric carbon, then we have no pre-available anomeric carbon to form. That sugar is called non-reducing. But if there is a pre-anomeric carbon, meaning that carbon 1 and then another carbon of the other sugar, not the anomeric carbon, then after the formation of glycosidic, we still have anomeric carbon that is free, whether it be carbon 1 in the case of aldosis or carbon 2 in the case of ketosis. So we have reducing sugar, reducing sugar when you have free anomeric carbon, non-reducing sugar when you have no free anomeric carbon. Example of free reducing sugar is maltose, lactose, and we will see from the structure. And the non-reducing sugar will have sucrose and trehalose. Of course, we also see how it responds from the stream. Okay, in maltose, what happened in maltose? Maltose is two glucose, meaning alpha-D glucose, comes together to form a disaccharide called maltose. So the glycosidic bond involved here or formed here is called alpha-1,4, meaning alpha-1,4, 1,4 meaning carbon 1 of one of the glucose and carbon 4 of the other glucose linked together. Because the carbon 1 is the anomeric carbon, and you have to indicate the configuration of that carbon. If the carbon is in beta, then it will be beta 1,4. If it is in alpha, it will be alpha 1,4. So because the configuration is in alpha, then alpha-D glucose combined with another glucose at carbon 4 to form alpha-1,4 glycosidic bond. And the reaction is basically a condensation leading to release of water to form maltose. This is a classical structure of maltose. So another thing, this maltose can be in the body, inside our system, can be broken down from the glucose monosaccharides, individual glucose unit, by an enzyme called maltase. So in a situation when the bond between the two glucose is alpha-1,6, meaning the anomeric carbon, number 1, and then carbon 6 of the other sugar, the disaccharide form is called isomaltose. And also, when the anomeric carbon is beta, not alpha, in beta 1 for the disaccharide name is called cellobiose. And what is cellobiose? Cellobiose is a disaccharide form during the hydrolysis or the breakdown of cellulose. You will see what is cellulose. Okay, the next one is lactose, still another reducing sugar. And what happened in lactose? Lactose is a disaccharide form between, or is, yes, disaccharide form between two sugar, beta D glucose and beta D galactose. And they are linked by beta 1 because the carbon configuration of carbon 1 or the glucose number 1 is beta. So we have the beta 1 4 glycoside. Say this is an example or another example of reducing sugar. The lactose is usually found in the milk. You know what is milk. So, and in them, the breakdown lactose is called lactose. Lactose is highly expressed. This enzyme is highly expressed in neonate, because the newborn children, they don't have, they don't eat anything that they've been fed from breast milk. So this enzyme is highly expressed, so that it can be able to break down the lactose to glucose and galactose. Glucose and galactose is now absorbed and then used for energy production in their body and drive the other metabolic processes. That is why you have high expression of this enzyme in neonate, the newborn, and as the child grows, you have the expression of this enzyme reduce. Sometime in adults, some adults, if you would have a very drastic decrease to the point that they become somehow intolerant to milk. I knew have experience of some people with lactose intolerance, that anytime they take milk, they used to develop some symptoms. So, yeah, intestinal discomfort, flatulence, etc., due to inability to break down lactose into. And this is just because their system drastically reduced the production of this enzyme called lactose. Now, okay. I thought that included the other one, sucrose. Sucrose, let me explain. So please, you can read about sucrose. Sucrose is another disaccharide formed between alpha D glucose and beta D fructose. You know, fructose is a ketosis. So they are linked together by alpha beta 1, 2. So we know that carbon 1 in glucose is an anomeric carbon configuration alpha. Carbon 2 in fructose, which is what? A ketosis. And carbon 2 in ketosis is anomeric carbon. Therefore, the bond is formed between an anomeric carbon in aldosis and the anomeric carbon ketosis, leading to formation of what we call the non-reducing disaccharide. Name alpha beta 1 4 or alpha 1 beta, alpha 1 2 beta. This is the type of bond. So the fructose, like I said, is non-reducing, and sucrose is the sugar that is being produced during photosynthesis by the plant. So when plant produces sucrose, they transport it to different storage part in the plant tissue, like a root, tuber, seed, and then it is stored in the form of that. Sucrose is very sweet, sweeter than lactose and maltose. And in fact, that is why it is even used as sweetener in the food industry. So the sugar we take, the table sugar we use, is sucrose made of alpha D glucose and beta D fructose. And sucrose is broken down in the intestine by an enzyme called sucrase. Sucrase releases the glucose and the fructose. So this is basically about disaccharide. So let's quickly look into polysaccharide. One thing important, and I need to say about polysaccharide is that one property of polysaccharide that distinguish it from other biopolymer like protein and nucleic acid. Remember, we said protein is a polymer of amino acid, nucleic acid is a polymer of nucleotide, which we already mentioned. We'll definitely see in the case of protein. So is that in the case of polysaccharide, it forms a branch due to the availability of the OH group that can easily form glycosidic. Remember, we said the glycosidic bond is formed between, it can form between carbon 1 to carbon 2 to carbon 3 to carbon 4 to carbon 5 up to carbon 6 of glucose. So all the OH group available there have the ability to form acidic bond. So you can have a branch. You can have carbon 1 forming a glycosidic bond with carbon 2 and or carbon, let's say carbon 4, and then the other carbon 1 form with carbon 4 like this. You have a long chain, but you have other carbon 6 can also react with another to form a glycosidic bond. You can have a long chain, and then it will have some branches. So which you cannot find in the case of protein, and of course you cannot find in the case of nucleic acid. I will give an example. In the case of nucleic acid, we made mention that one nucleotide is attached to other through the 5' carbon or 3' carbon ribose. So you have one attached to, you have a phosphate group attached to 5', then of the other, the phosphate group of a new or another nucleotide will attach to the nucleotide is to be formed, will attach to the 3' like this. So you can see the 3 and the 5' are all occupied in the nucleotide. So there is no any other point forming the what we call phosphodiester bond. So also in the case of protein, we have the formation of dipeptide bond, which involves the amino group and the carboxyl group, which shortly we'll discuss that, I think, in the next, should be in the next week class. So we'll discuss that definitely. So, now the polysaccharide. Having to know one of the difference, we also need to know that these polysaccharides are so diverse. So diverse. How do they differ from each other? There are a number of points where they differ from each other, like in the identity of the recurrent monosaccharides. We mentioned many monosaccharides, so you can have the polysaccharide that contain more than one monosaccharide, a different one, multiple. And you can have one that is straight containing only glucose attached to another glucose attached to another glucose like this. Another point is that the length of the chain, you can have a very short, some of them are very lengthy. The type of bond, we mentioned different types of bond, alpha 1, 4, alpha 1, 6, alpha 1, 3, alpha 1, 2, so also they determine which carbohydrate is. And the degree of branching, some of them are not even, doesn't have branch, some of them are branch, highly branched, so the degree of branching. So basically this polysaccharide are classified into homopolysaccharide. In homopolysaccharide, you have polysaccharide that contain a single type of monosaccharide, while in heteropolysaccharide, you can have polysaccharide that contain 2 or more different kinds of monosaccharide. When it is a homopolysaccharide, you can have a polymer that contains only glucose. It's generally called glucans. If it is mannose, it is called mannans. If it is galactose, it's called galactans, like this. They are also further classified into Okay, example of homopolysaccharide like, okay, I've already mentioned galactosan, mannans, fructosan, glucans, etc. And can also be classified into storage or structural. In storage, we have monosaccharide that is been, polysaccharide that is been stored in the body for energy, maybe for energy production, while in the case of structural, for structural function like cell wall or animal exoskeletons, which we will see example in cellulose and chitin. So now let's quickly look at each of these classification, starting with the homopolysaccharide. In homopolysaccharide, we have example homopolysaccharide and consider a storage polysaccharide, glycogen. Glycogen is a storage polysaccharide. So animals store polysaccharide or monosaccharide or sugar in form of polysaccharide called glycogen. Glycogen usually is the polymer of glucose linked by alpha-1,4 glycosidic bond. In animal cells, glycogen is usually stored in skeletal muscles and the liver. So basically, liver and then skeletal muscles. So it's a polymer of glucose linked by alpha-1,4. It is highly branched in the sense that the branch occur after every 8 to 12 residue. So after you count 8 residue, you can see a branch. Or 12 residue, you can see a branch. After every 8 or 12 residue, you will see a branch. And at the branch, the link or the glycosidic form is alpha-1,6. So the molecular weight reaches up to several millions. Like we mentioned, it's function mainly as the storage polysaccharide. And somebody will wonder, why wouldn't the glucose or the monosaccharide, as it's just maybe it's a polymer of glucose, wouldn't the body store the glucose instead of converting it to glycogen? Yeah, of course it can, but it is not possible. Simply because, so now the question is, why store glucose in form of glycogen? Now, the reason is, when the body decide, or for example, yes, the body decide to store glucose in form of glucose or sugar in form of glucose instead of glycogen. The concentration of the glucose, like concentration up to like 0.4 molar. And you see 0.4 molar is highly concentrated. And which, if converted to glycogen, can only form 0.01 micromolar. So now just imagine a cell containing substance that is up to 0.4 molar, highly concentrated. And we know the process of osmosis, the movement of water region, high concentration. So definitely we'll have water going into the cells until the cell is filled up with water to the point that the cell can burst. Once it bursts, then that's a problem. So to avoid that increase in the concentration, the osmolarity, it is converted to glycogen. And if that 0.4 molar glucose is converted to glycogen, it will only form 0.01 micromolar, which is insignificant. Now, the concentration of the glucose will show at a point that you can even have the concentration of the glucose in the extracellular fluid outside being high. Therefore, so when you eat a food, this glucose can move in. But it has been the glucose exists in form of glucose inside the cell. So even when you eat food, the concentration of the glucose inside is higher than outside. So the movement of the glucose is the one that comes from the food. Very difficult, it cannot go inside, meaning going against the concentration, which is not possible. Rather, you can even have some of them coming out. So another one we just mentioned that we could. First one we say can lead to bursting of the cells. Number two, we said it can prevent the glucose uptake into the cells. So the next one is starch. So starch is a storage polysaccharide plant. It's also a polymer of alpha D glucose, just like glycogen. So starch is linked by alpha 1,4. So starch comprises of two polymers. Amylose, which is a linear polymer of glucose, doesn't have any branch. And then amylopectin. The amylopectin is branching chain that contain a branch at alpha 1. It's just like a glycogen. The only difference here with amylopectin and the glycogen is that the glycogen is highly branched. So the branch is high in the glycogen when compared to amylopectin because the branch occur after every 24 to 30 sugar residues instead of the 8 to 12 residues as in the case of glycogen. And, we said starch is broken down when it would to contain starch. What happens is that alpha amylase, pancreatic amylase is produced from the saliva, and the one produced the salivary amylase produced in the mouth, and then pancreatic amylase produced in the stomach. And the ones that is to break down starch. And the same enzyme break down glycogen, whatever they are needed. So the molecular weight of amylopectin is up to 2.3. So the next class of let me talk about that. Another one is dextran, which is a homopolymer also. The homopolymer dextran is produced by bacteria or yeast. It is made out of the glucose linked by alpha 1,4, but it has a branch, and the branch can occur in alpha 1,3, alpha 1,2, and alpha 1,4. So this is basically dextran. So another homopolysaccharide that are considered structural now, not storage, is cellulose. Cellulose is a polymer of beta D glucose. Now, beta, not alpha, beta D glucose. And cellulose is the fibrous and the water insoluble substance that made out of the plant cell wall, like the cocoon, the stem, the trunk, the woody part of the plant. The grasses and the rest contain a lot of cellulose. So this cellulose is a linear, unbranched polymer of glucose, like we said, linked by beta 1,4. It doesn't have any branch. So the cellulose cannot be broken down by alpha amylase. Alpha amylase can only break down a polymer linked by alpha 1. But this one is beta 1. And the humans do not have this enzyme, cellulose, the enzyme that breaks down cellulose. Therefore, we cannot eat the grasses. We can, even when you eat the grasses, you will etiquette its grasses. But only ferment existence. And if it ferments, just like as animals do, animal eats. Animals also do not, like goats and the rest, do not have this enzyme. But what happens because they have a stomach called rumen, and the rumen contain a lot of bacteria. So those bacteria can release the cellulose enzyme, which break down this cellulose to form some glucose or other things that can be utilized by bacteria. So another one is, before I talked about chitin, this is the heteropolysaccharide. I will give another example of homopolysaccharide that is also structural, like chitin. Chitin is a linear polymer of beta N-acetylglucosamine. Remember, I already told you what is N-acetylglucosamine. Usually it forms the exoskeleton of insect. Of course, insect doesn't have a bone, but they have an exoskeleton. What is made up of that exoskeleton? That exoskeleton is made up of chitin, which is chitin is a good linear polymer of beta N-acetylglucosamine. Now, the heteropolysaccharide. The heteropolysaccharide example, we have agar. We say the agar is a branch heteropolysaccharide composed of two polymer, agarose and agarophotin. Agarose is unbranched, it's linear, does not have the agarophotin is branch. And we said that agar is a polymer of sulfated D and L galactose. Derivatives meaning the sulfated form of D-galactose, the sulfated form of L-galactose, linked by ether link at carbon 3 and carbon 6. We say agar serves as a component of cellular lipids. We already mentioned that it's made up of the linear agarose and then agarophotin, which is branch. So we also have glycose aminoglycans. Glycose aminoglycans also heteropolysaccharide, the structural function. And how do I explain this? Remember, I talked about, when I was talking about organization of life, I mentioned that from cells, we have tissues. Tissues comes together to give, cells come together to give a tissue. How do these cells come together? What holds the cells together to give a tissue? That's the question. So we have this, they form the extracellular matrix. So they are responsible in holding the cells. They form a kind of gel-like material that hold the cells together. In addition to some structural protein, fibrous protein like collagen, elastin. So they are the one that is responsible in holding the cells together. So they form the extracellular matrix of the cells. So once something go inside the cells, it comes from the extracellular, and the extracellular is made up of all this stuff. So glycose aminoglycans, they are linear polymer of a repeating disaccharide. We have been talking about a polymer containing monosaccharide, but this one you have disaccharide, and then the same disaccharide like this formed a long chain polymer. For example, one of the disaccharide is usually N-acetyl glucosamine or N-acetyl galactosamine. And then another one will be a uronic acid. A uronic acid or a sulfated or a sulfur attached to uronic acid. For example, there are many. For example, we have hyaluronic acid. Hyaluronic acid is a polymer of glucuronic, it is the disaccharide unit, glucuronic acid, and N-acetyl glucosamine. So you have a disaccharide of glucuronic acid and N-acetyl glucosamine that keep on repeating themselves to form a long linear polymer we call hyaluronic acid. Like chondroitin sulfate is glucuronic acid and sulfated N-acetyl galactosamine. Another one like heparin, which is a sulfated glucosamine, which is sulfated glucosamine plus a sulfated glucuronic acid. So a linear form of this disaccharide gives rise to what we call alphabet. And we have some of them here, which I mentioned glucuronic acid. I've already mentioned all this. And they are usually found in different systems like hyaluronic acid, serve as lubricant, shock absorber, chondroitin sulfate, maintaining the structure of the tissue, like heparin as anticoagulant, which I think you're very much familiar with in the clinical terminology. We have dermatan sulfate, which maintain the shape of tissues, and keratin sulfate usually found around the eye cornea, and it keeps the cornea transparent. So I think if there is anyone that we didn't discuss is peptidoglycan. Peptidoglycan is also a heteropolysaccharide which forms a polymer of N-acetylglucosamine, which I've already explained, and N-acetylmuramic acid, I've already explained, linked by beta 1,4. So this peptidoglycan form the component of bacterial cell wall. We know what is bacteria. So this polymer of N-acetylglucosamine and N-acetylmuramic acid linked by some short peptides to make it very strong, form a cover to the bacterial cells, thereby making the bacterial cells to survive inside system, inside our body system. For example, you have water aqueous solutions that can easily go into the bacterial cells and kill the cells. But because this polymer of N-acetylglucosamine and N-acetylmuramic acid peptidoglycan, with the help of this short peptide that bring them together, form a coverage where you have the cells hiding between this peptidoglycan. So the water cannot go in, the solution cannot go into the cells and rupture it. And I think one of the mechanisms of even antibiotics like penicillin is to prevent the synthesis of this peptide that form the cross-link. So that once the peptide cross-link does not form, the bacterial cell will become porous. So substance can easily go in and develop the cells and dilute the base of the cell, basically into penicillin protein. So if you have any question, please do not hesitate. I will try and ensure that I'm taking this to the first aid and then I'll try to take your time. If you have a question, please let us discuss.